Corals are happiest and healthiest where the water is clear and well lit by the sun, and where they don't get too much competition from seaweed. But their unusual lifestyle makes them especially vulnerable to a changing environment.
Corals are actually animals, but they're glued to the ground like a plant is. They're not able to pack their bags and move to another side if the conditions is no good for them.
Where the carbon dioxide bubbles are most intense, few corals seem to be finding the conditions favorable. These large, extremely tolerant corals are called porites. They have thick skins and now dominate the reef. The colorful branching corals and soft corals that flourish in most coral reefs are gone. The team works around the clock in all kinds of weather. Some days, nothing goes according to plan.
I think there's some problem with one of the generators. One of the exhaust pipes had a bit of a breakdown.
Underwater conditions are difficult.
This site we have massive rocks, so it's really hard to drill inside the structure.
And some of the coral experiments have become dinner for some hungry snails.
At least 50% of little coral branches we made had been eaten by these snails.
Yeah, not happy. Experiments by nature quite often fail. It's requiring a lot of patience. But eventually we'll sort it out and we'll find some other ways to test the same questions.
While Katerina is studying the impact of high acidity on the corals themselves,
So I probably first jump in and
another team member, Laetitia Plaisance, is studying the many other sea creatures who depend on them. Where the water chemistry is normal, the coral gardens of Papua New Guinea are among the richest on the planet. Home to more than 500 species of coral. And hundreds of thousands of different kinds of tiny sea creatures. The entire ecosystem rests on a coral foundation built from calcium carbonate.
everything is a balance in the ocean. The reef is made of calcifying organisms, so that means they need higher pH to be able to build their skeleton. And this skeleton then provides the structure of the reef where all the other associated species can find shelter and they can mate and they can feed, they can hide in that structure.
Today, Laetitia is focusing on one particular habitat, the dead coral head.
I'm on my way to find a coral head, a nice dead coral head.
Living coral heads provide homes for tiny fish and many other animals. Even on normal, healthy reefs, coral heads will eventually die, but their calcium carbonate structure remains. And, just like a dead tree, new creatures move in. Laetitia collects dead coral heads from both the normal site and the bubbly site, where acidity is much higher and the pH is lower. Will she find the same variety of species living in both locations? To find out, she cracks open the coral heads, hunting for the creatures hidden inside their nooks and crannies.
I'm searching for the crabs and the mollusk, and here is a little ophiurid trying to escape. Another sea cucumber, I think, here. All part of the food chain, and if you miss a link in the chain, the rest of the chain can continue, can't go on. So yes, all those little, pretty insignificant species are extremely important.
When she takes apart the coral head from the control site with normal pH and acidity, she's impressed by the number of creatures she uncovers.
This morning we collected a nice dead coral head from the control site, which meant regular condition. It was a beautiful reef, and when I opened the coral head, it was filled with a life, abundance, and diversity. So we have plenty of different species of crustaceans here, squat lobsters and crabs and shrimps.
But examining the coral head from the bubble site, even before Leticia breaks it open, she is concerned.
The first thing that I noticed with this coral head is that it's covered with this type of algae. And the problem really is that this type of algae does not hold much diversity, which I'm used to see in other sites where the pH is much higher.
The collection of life looks more limited, but to make sure, to get an exact inventory of all the different species, Leticia analyzes the DNA of the samples she collected. For example, two tiny shrimp may look alike, but they could actually be different species. A DNA test will reveal precisely how many different species live here. The DNA results confirm what Leticia suspected. The highest species diversity is at the control site, where the acidity and pH are normal. But as the acidity goes up and the pH goes down, the number of species drops dramatically. For each type of animal, only one or two species are tolerating the high acidity, and the rest have disappeared.
So you have the same kind of shrimp 100 times, when in the regular pH conditions you have 100 different species.
Leticia finds that the high CO2 site has 30% less diversity of small sea creatures, like hermit crabs, shrimp, and starfish, than the control site. For Leticia, this is troubling news.
The reason why this group is very important is because it's the basis of the food chain in coral reef. The fish feed on those tiny creatures. So if those tiny creatures disappear, the fish community is going to change and evolve as a result of this loss.
It appears that changes in acidity aren't just affecting the corals themselves, but hundreds of species living at the bottom of the food chain. And if those at the base of the food chain are at risk, so are all the creatures that rely on them for survival. These kinds of changes aren't just happening at coral reefs. Cold water absorbs more carbon dioxide than warm, so acidity could rise in the Arctic and Antarctic seas more quickly. And that could mean trouble for the creatures here that, like coral and oysters, rely on carbonate to build their shells and skeletons. Aboard the Aurora Australis, the Australian Antarctic Division's research vessel, scientists are studying tiny plankton, some the size of a pinhead.
So we get to throw that back over the side.
Donna Roberts has spent much of her career in the Southern Ocean, and her favorite kind of plankton are tiny animals called pteropods. These almost transparent sea snails swarm in vast numbers in the colder waters of the Arctic and the Antarctic oceans. They are at the bottom of the food chain, providing food for whales, fish, and ultimately us.
The pteropod shells are gorgeous. It's just like a garden snail, but instead of having one slimy foot, it's evolved to live in the ocean with wings. Scientists call pteropods the potato chips of the sea because they're meaty and a lot of things eat them, but they've got that added little crunch.
Tucked away in this cold room at the University of Tasmania are pteropod samples from Antarctica, dating back to 1997. Donna is analyzing their shell weight to see if rising acidity has affected them.
it does matter if a little organism is losing weight because it's a measure of how healthy they are. So if their shells become thin, then they're going to put more of their energy towards building what little shell they can than reproducing.
Pteropod shells collected today are 35% smaller and more fragile than those caught a decade ago. Yet another sign of rising ocean acidity.
So this is a healthy set of shells that we collected in 2002, and I'll just show you for comparison what unhealthy shells look like. So you can see this one here has lost a segment of shell, and this one here is incredibly thin, very paper thin. Shells like this often fall apart in my hand when I'm trying to weigh them.
Scientists say that, like the baby oysters, there could soon come a point at which the levels of carbon dioxide will be too great for these beautiful little sea butterflies.
Most people have not heard of what a pteropod is, but they are vastly important. They're holding up the entire Southern Ocean food web in places.
Pteropods are a key food source for lots of animals that swim the seas. Not only in the southern oceans, but in the northern oceans as well. So what would happen if pteropod populations decline, or even disappear, around the globe because of rising acidity?
The ecosystem effects of removing all the pteropods are just completely unclear. If we take them all away, we really don't know what the effects might be, but they may be quite dire for the things that feed on them.
Among the things that feed on pteropods are things we feed on, like salmon.
If you're a pink salmon, you can't just remove 10-15% of a juvenile pink salmon's diet and expect it to, I'll just eat a little more of this instead. It's not evolutionarily hardwired to do that. So when you start removing huge components of what other individuals eat, what other species eat, that's a problem.
If higher acidity reduces or wipes out populations of creatures at the bottom of the food chain, the consequences could be bad for those further up. The worst possible scenario would involve a chain reaction of extinction, resulting in mass die-offs throughout the oceans. It has happened before. The fossils that trace the history of life in the ancient oceans reveal dramatic moments when suddenly many living things went extinct.
We think many of the big extinction events in the past have been related to some rapid event, whether it was an asteroid impact or some rapid, large increase in volcanism. Temperature was changing, ocean chemistry was changing. That's where we see the big extinction events.
The most famous extinction was 65 million years ago, when the dinosaurs disappeared. But an even bigger catastrophe hit the oceans 250 million years ago, when the Permian era ended. The leading theory is that massive volcanic eruptions caused carbon dioxide levels to spike quickly. The result was what's now known as the Great Dying, when up to 96% of all marine life went extinct, including many coral reefs. If acidity changes slowly, many creatures may be able to adapt and evolve to cope with the new conditions. But sudden changes are often dangerous for life.
On evolutionary timescales, organisms are adapting to their environment. The environment's changing and life is always changing. The more rapid that change, the harder it is for the organisms to adapt.
And today, the burning of fossil fuels is releasing so much carbon dioxide, it's changing the environment of the ocean very rapidly. Scientists fear that if carbon dioxide continues to rise at high speeds, we could have a major extinction event on our hands.
The rate of change is happening much, much faster than these plants and these animals can adapt. Unless it happens at a much, much slower rate, evolution will not have an opportunity to keep up with the change.
Perhaps nowhere on Earth are the plants and animals of the sea more at risk than on coral reefs. 500 million people rely on these unique ecosystems for their survival. The fish that live around reefs form a major part of the human diet. So, how will rising ocean acidity affect these animals? In addition to messing up the base of the food chain, will the changing chemistry have any direct effect on the fish's own bodies? Researchers like Danielle Dixon are trying to find out. Until now, most of her work has been in the lab, but today she's joining the team in Papua New Guinea, where at one unique reef, carbon dioxide bubbles are raising the water's acidity to extreme levels.
The thing about this location that makes it really, really interesting is that these fish have been living in this CO2 situation for a very long time.
You can see where the CO2's coming up there. Yeah, so that's the CO2 seep site.
Philip Munday is Danielle's former professor and collaborator. In experiments back at the lab, they exposed fish to high levels of CO2 and saw alarming results. The damage wasn't done to the fish's skeletons, but to their brain chemistry and their behavior.
What we do see that's really dramatic is the changes in their behaviour. So things like how they respond to chemical cues in the water changes, their response to sound changes. And what's really cool here is that we can come to these seeps, we can see fish that have probably been living for months, maybe even years under a high CO2 environment and see if their behaviour is still affected.
The team wants to find out if the behavior they saw in the lab will be repeated in the wild, where some fish have grown up in high CO2 conditions throughout their lives. They'll compare the behavior of the fish living both near and far from the carbon dioxide bubbles. First, they'll test how they respond to chemical cues in the water. In other words, their sense of smell.
Baby fish in particular have very good noses. It's not on the end of their face, but they have nostrils and water comes in and there's sensory cells inside the nostril and then water goes back out. Very much the same as we do.
Just as land animals use their noses to detect certain molecules that float in the air, fish noses can sense molecules moving through the water. Their sense of smell is critical for their survival. They use it to catch food, find their way home, and even to sense danger.
There's a lot of things in the water that produce a lot of smell. There's predators in the water that are trying to feed. There's prey in the water that things are trying to feed on. The corals themselves, they're also releasing chemical cues. Pretty much everything uses chemistry underwater. So, reefs would be a pretty smelly place.
The first step for this experiment is to catch some fish.
They're very easy to catch. Just a little bit of club oil, and then you can use your hand to waft them out of the coral, grab them in a net.
Next, they must catch a predator fish.
So I got a fish.
Danielle wants to test the smaller reef fish's ability to identify their predators, like this rock cod, by their smell. A crucial survival strategy for animals on land or sea.
This fish is going to be used to make predator odor. I'm just going to soak it in some water, and then the cues from the fish will come out in the water, and I'll be able to test little fish to see if they can identify the predator cues over other cues.
For this experiment, water with the predator odor is being pumped into the top side of the flume. The bottom side has normal offshore seawater.
The fish that we have in the flume right now is taken from the control reef where no CO2 bubbles are happening. The fish identifies the predator through smell alone and stays in the offshore water side where no predator smell is.
The next fish has grown up in the carbon dioxide bubble site, where the water acidity is higher.
Now the fish is sitting in the water that has the predator scent.
Instead of being repelled, the fish seems attracted to the dangerous smell.
We've found that the CO2 is affecting the fish's cognitive ability, so it can identify the smell of a predator, but something's wrong with its brain that it's having a hard time identifying it as a bad thing. It's just identifying the smell.
After testing dozens of fish, Danielle finds that the fish that grow up in higher acidity don't avoid the predator smell, behavior that makes it much more likely that they'd end up as somebody's dinner. In addition to smell, Danielle wants to test another survival instinct, the ability to hide. On these reefs, many fish live among the corals, using their branches for protection.
These fish are like candy on the reef. They really like to be eaten. It is in their best interest to be closer to their habitat so that they can retreat back to it and they're not picked off as much by predators.
Danielle tracks how adventurous the little fish are, which ones will play it safe and stick close to the coral shelter. This one is from the control site, where the conditions are normal.
You can see it retreats right into the coral. It might be exploring the habitat of the coral, but it's not really exploring the tank. The fish will stay like this for the entire trial in general.
The next fish comes from the carbon dioxide bubble site.
You can see it's checking out the water a lot. It's going quite far from the coral. It's not really retreating back into the coral. Being bolder could make you grow faster and get bigger, but it also makes you a lot more susceptible to predators.
Danielle tests several fish. The small fish living in the higher acidity are consistently more adventurous, behavior that could put their lives at higher risk. Researchers don't know yet how all the different kinds of fish that live in the sea will be affected in higher acidity. Some are likely to cope better than others, and some may find some surprising benefits. At the bubble site, one form of life that's thriving is the seagrass.
Seagrasses can live in very high CO2 environments. They actually like it because seagrasses, like the terrestrial plants, they do take in the carbon dioxide and convert it into sugars.
For plants, carbon dioxide is a fuel. They use sunlight to turn it into sugar, leaving oxygen as a waste product. The rich supply of carbon dioxide here is fertilizing these seagrass beds. The thick lawn of seagrass appears to be an important nursery for baby fish. Biologists are hoping that many of today's fish will find a way to survive in an ocean with more carbon dioxide and higher acidity. Their bodies may be able to adapt to changing conditions, possibly by turning on particular genes.
There are mechanisms that might allow them to adjust to a high CO2 world. Some individuals will have better genes for that particular environment, so they pass their genes on and you get genetic adaptation.
Marine biologists are eagerly hunting for any signs of genetic adaptation to a rapidly acidifying ocean. Today, the race is on to find creatures that carry genes that would help them survive. And the best places to look are the ones where ocean acidity goes